Good morning again to those that have joined us on the web. We will continue now with the student presentation, and the first one will be Habte Nida from Ethiopia. Habte graduated with the Bachelor of Science degree in Plant Sciences from University of Hawassa in Ethiopia. He has been working as an assistant researcher in Werer Agricultural Research Center, which belongs to the Ethiopian Institute of Agricultural Research, and he was there involved in areas of crop improvement. And Habte will give us a talk on the investigation of genetic loci involved in reproductive heterosis in sorghum bicolor. Habte, please.
Good morning, everybody. Thank you, Professor Saranga. The title of my presentation is Investigation of Genetic Locus Involved in Reproductive Heterosis in Sorghum Bicolor, conducted under the supervision of my advisor, Dr. Fridman.
Will work now. Give it 2 seconds to. Okay.
Thank you.
It's yours. Sorry.
So again, the title of presentation is displayed here: Investigation of Genetic Locus Involved in Reproductive Heterosis in Sorghum Bicolor. So to define heterosis or hybrid vigor, it is the phenotypic superiority of a hybrid over its inbred parents. Here this picture shows that this picture was taken from our experiment this season. So this the plant in the middle is a hybrid, and this one is one of the parent and the other parent. So you see here clearly the variation between these three genotypes. So the question behind this one is so far there is no answer for this. I mean, the genetic mechanism behind this phenomena is not known. So there are models of heterosis still currently used. So the first model is the dominance model, in which recessive alleles are different loci complemented in the hybrid. So in the illustration here you see you have a recessive small a and small b coming from or inherited from two parents, P1 and P2, and you have also a dominant favorable allele B, capital B and capital A coming from P1 and P2. So in the hybrid, the two recessive deleterious alleles will be complemented by the two dominant alleles. So this is the hypothesis supporting the dominance, and it was developed in 1808 by Davenport. The other hypothesis opposing this idea is that you have here the illustration here. You have a homozygous situation in both parents in one of the chromosomes from P1 and P2, but you have a different allele combination. So you have here B asterisk and B asterisk in one of the parent, but B in the other parent. So this is assuming that there is no dominant relationship between the two. That means between B asterisk and B. So in this case, you will have intra-locus interactions. That means interaction between the two locus. So in this case, you will have superiority of the hybrid over its both of its homozygotes. And this idea was independently developed in 1808 by Shell and East. another is a kind of genetic intermediate between the two. So the pseudo overdominance refers, you have here the illustrations as there is tightly linked dominant alleles with recessive deleterious alleles in both parents. So these are tightly linked in repulsion or in transarrangement. So in the hybrid, they appear to be like an overdominant mode of inheritance, and you will have advantage from this combination. But truly, this is the dominant case. So these are two of these are the major models of heterosis known so far, and this is a genetic intermediate, but truly is a dominant one. These are some of the examples. Contribution of overdominance heterosis in males by Liu et al. In 2003. Again, overdominance for reproductive traits on a population of tomato interrogation lines. In this study, they have showed that they identified other forms of gene action other than overdominance for other traits, but they identified overdominance mode of inheritance for reproductive traits in tomato interrogation lines. Again, another study on maize immortalized F2 population. They correlate heterosis with partial dominance and overdominance for plant height in maize. This is Jihua et al. In 2007. Again, another study. This is from a recent finding that brought by Gore et al. And Macmillan et al. So from their investigation, they say that pericentromeric regions may contribute to heterosis. This region is known for low recombination rate, and maize also genome. They show that maize genome contains many pericentromeric regions. So this region has residual heterozygosity. So because of this, they propose that this region may contribute to heterosis as well. Another recent example from a single gene mutation. This is the most profound example done in 2010 by Krieger et al. So they show that a single mutation or a single heterozygous gene may confer heterosis in tomato. So let's see now how the genetic loci were detected or developed in Israeli sorghum core collection by a PhD student in our lab. It was done by Imri Ben Israel. He is a PhD student in our lab. So he did a dialysis scheme and a different set of approach. So if you see this, the first three and the first two, they used F2 or interrogation lines. So they used mapping populations to generate or to scan the genome or a genomic scan using mapping population. This was a mutation. So this is a taught from a pericentromic region. So that means a contribution of this region for heterosis. But in this approach, the idea is genomic scan, but the approach is a bit different. Here, you have, initially there were about 200 sorghum collections which represent sorghum from throughout the world. So they were studied for polymorphism using 51 SSR markers. Then 20 founder lines were selected based on their diversity and relatedness. You have here founder line 1, 2, up to 20. So these are selected based on their diversity and also relatedness. Then after that, they were crossed in a diallel scheme. Here you have the hybrids 1 and 2. For instance, you have hybrid between 1 and 2, so you will have a hybrid here with a heterozygous situation, one chromosome coming from one of the parent or one of founder line, and the other from other founder line. So you will have here heterozygosity, or in some case you may have homozygous locus. In some case you will have heterozygous locus. So this is continuing up to hybrid 1920, so all diallel scheme. Then after crossing this, overdominant heterosis was calculated using this formula. So if the hybrid is between the two parents, you will have an overdominant heterosis value of 0. or else if you have the hybrid is either higher than both parent or lower than both parent, you will have a positive and a negative over dominance heterosis value. So based on this value, mean of ODH value of each of the plant is calculated, and the mean is calculated from each plant for each hybrid. So after that, a genomic scan and association of over-dominance heterosis with heterozygosity has been conducted. Here you see that if you see locus X, you will have individuals. These dots represent each of the hybrids. So you will have this hybrid containing the heterozygous locus where having the high ODH value. This is in a y-axis is the level of heterosis, and you have here the homozygotes and heterozygotes. So in locus X, you see that the heterozygotes with this allelic combination were significantly higher and strongly associated with the level of over-dominance heterosis. So this was the approach to develop heterotic locus. In other locus, in locus Y, more or less you see very uniform level of heterosis between the heterozygotes. So this is the approach. So after that, here you see on chromosome 4, one locus was strongly associated with heterosis in 2010, and also it has been confirmed also in 2011. So then after looking the significant marker here, then a scan for additional markers. That this is zooming in on the left, no, on the right. So here you see that in this region, most probably you will have some kind of genes or gene families contributing for heterosis. So you see here on chromosome 4, in 2010, it was significant. One of the markers was significant. Then looking for additional marker, additional significant marker was identified in 2010 also. So therefore, in this region, there is some kind of gene mediating heterosis. So our assumption was to test this marker. So this marker was the marker that I tested in F2 population. This was in F1. So the objective or the hypothesis was this marker should behave as heterotic in F2 population as well. If it is heterotic in F1, it has to be heterotic in F2 as well. So we have to see the contribution also. So the objective was to verify this heterotic locus in F2 population, and also with specific objectives of developing genetic markers, defining HTTL for genotyping, genetic analysis of parental lines and derived F2 populations, analyze genotype, phenotype of selected markers, as well as a separate study, we did a study of to characterize and define sorghum panicle development. So 240 F2 population of each of the crops were planted in rows along with their parental line and their hybrids. And planting was arranged in 3 blocks, each of the blocks containing about 1/3 of the populations, and randomly spread 3 replicates of parental lines and F1 hybrids. So out of this, DNA extract from F2 parent and F1 hybrids using standard method, and a total of 204 and 147 F2 plants were genotyped for the candidate locus. And out of these, 170 and 146 of the genotyped F2 plants were considered for genotyping and for further analysis. Some plants were discarded due to poor establishment in the experimental site. So the methodology used to genotype the marker was, here you see relatively a new approach. It was originally supposed to be separated on a high resolution MELT analysis. Here you see this is a relative level of fluorescence here on a y-axis and melting temperature. So whenever you have a different amplicon, you will have a different pattern of melting curve. So this was supposed to be, I mean, our genotype were supposed to be separated on a high resolution meta-analysis, but we couldn't get consistent and reliable result from this one, so we used gel to separate the amplicon, so it does pretty well. Here you see the two homozygotes. This is parental 1 and parental 1, and the two are parental 2. That means look like the other two parents, and this is a hybrid. So it was separated on a high percentage gel. Data on plant height, stem diameter, fresh leaf weight, and stem weight, as well as other traits measured as an agronomic trait. And on panicle also, dry panicle weight was measured after oven drying for 2 days at 70 degrees, and also panicle length measured from the same panicle. Beyond this, we go into detail of the panicle architecture, like primary branch number, primary branch length, secondary and tertiary branch number, 100 seed weights. This is because there is extensive branching nature in sorghum, and to see which of the inflorescence characteristics or sorghum architecture is contributing for yield, as well as to see the multiplicative effect of the different traits coming from the different parent. So we did a dissection experiment on this also. So here you see the sorghum panicle. We have here the primary branch. These are the primary branch. Then in bottom third, in the long branch zone, according to Brown et al., we selected two primary branches randomly. Then from each of these two, we calculated secondary and tertiary branch, as well as tertiary and secondary branch, as well as we measured the length of the two branches. Then finally, mean of the two branches were used to characterize each of the panicles. And also, finally, the panicle was traced to determine 100 seed weight as well. On a different experiment to see the shoot apex development of sorghum, we did another experiment. Here we see starting around 28 days after sowing, there was a transition from vegetative to reproductive structure. Here you see the picture, a dome-shaped apex growing. So after that, we sampled 4 plants every 4 days from 2 embryo lines, so 2 founder lines. So here you see developing sorghum panicles and measuring it with a specified scale, and later we analyze it for the length of the apex. Data on test on chi-square distribution of the F2 genotype, analysis of variance on agronomic and panicle traits, as well as calculation of overdominance heterosis, as well as percent retained in F2 population was done. So let's see now what we identified from our experiment. This is a genotype segregation at F2. So you see that in both populations, this is in population 1 and this is in population 2, 2 crosses. In both crosses, there is no significant difference between, according to the chi-square distribution, you have a 1-to-1 distribution. So the population was segregated according to the chi-square distribution. This is a plant height in both populations. This is cross 1 and cross 2. Here on a y-axis is a plant height, and an axis is the parent 1, F1, the parent 2, and the 3 F2 population. So here you see there is heterosis for plant height as well. Parent 1 seems to contribute for high parent alleles. In this case also a bit is better as compared to the second parent and the third parent. So there is heterosis for plant height. But there was no significant difference between the 3 genotypes. You have the homozygote 1, parental type 1, and the heterozygote for the candidate locus, and the homozygote 2, this is parental type 2, but there is no significant difference in terms of plant height. Again, there is also no significant difference between the two. Plant P1, that means a parental type 1, seems to contribute for more of the high parent alleles. This is a stem diameter. Again, there is a heterosis for stem diameter as well. This is parent 1, and this is parent 2, and F1 in the middle. So stem diameter also increased with the heterosis level, but there was no significant difference between the three genotypes. This is a fresh leaf weight. Again, you see clearly difference heterosis in terms of in F1. You see here, this is parental type 1 and parental type 2. Again, a small increase probably, but it was not statically different between the three genotypes in fresh weight. And here also more or less is similar. The two parents on left side a bit lower as compared to the F2s, but there was no much significant difference in the second cross. But you see clearly there is a heterosis for fresh leaf weight as well. This is a stem weight. Again, there is much significant heterosis in F1. This is parental type 1 and 2, but there was no significant difference between the three genotypes in F2. Similar in the second cross also. But here we see that there is a bit interesting result here. This is a dry panicle weight of the three genotypes, genotype for the candidate locus. You have the homozygote 1 on left side and homo 2 on the side. This is for the two cross. This is cross 1 and cross 2. You have the dry panicle on a y-axis. So you see that there is some level of heterosis due to heterozygosity as single locus. Here you have about 23 and 10% increase in dry panicle weight due to heterozygosity at a single locus. So if you agree with me with the probability level, 10% probability level probably, so there may be contribution from this locus. So in both populations you see there is an increase here in 12 and 17% increase in dry weight as compared to the two homozygotes. This is a dry panicle weight of the parent 1, parent 2, and clearly seen heterosis level, or there is a huge increase in dry panicle weight in F1. Again, dry panicle weight, this is in comparison to the F1 and the other parent as well. Here you see that there is also this variation in between the three genotypes. So the heterozygote tend to be higher as compared to the two homozygotes. Maybe this is even it indicates that there is significant difference as compared to parent 1 type marker, but maybe statically it's similar to this one. In this case also, in the second cross, you see the heterozygote was higher than the two homozygotes. This is dry panicle weight over dominance. Again, in F1 you have around 100 over dominance, whereas the 50% top plants, this is to show that whether heterosis was mediated by a single diallelic gene. So you have here the top 50% of the plants resulted about 60% of the F1 heterosis, whereas the heterozygote again is slightly higher as compared to the two homozygotes. F2 was around 25% overall. So this indicates that much of the F1 heterosis was not retained in F2 population, which indicates that so many genes are involved in heterosis. This is the retention of F1 heterosis. In F2, you see so here about 60% of the heterosis was retained by the top 50% of the plants, whereas about 32% of the F1 heterosis was retained by the heterozygote for the candidate locus. So you see there is variation between homozygote 1 and homozygote 2 in terms of retaining the F1 heterosis. So probably you may say that there is some level of heterosis or advantage due to heterozygosity at the candidate locus. So you see that top 50% of the plants should maintain or most of or 100% of the F1 heterosis. You see here, I have tested this one statically. They should maintain about most or 100% of the F1 heterosis. If a single gene was to mediate this, but it was not true, and it was only 60% of the F1 heterosis was maintained by the top F2 plants. This is a panicle length. So you see here, there is no heterosis for panicle length. The hybrid was more or less similar to the second parent. So panicle length may not be contributing for heterosis. So you see that again there is the hybrid is not included in the second cross. Probably it would be the same as the other parent. We don't know. The parent one is a bit shorter in panicle length, but there was no significant difference between the three F2 genotypes. Okay, now let's see the panicle traits. So which of the panicle traits are contributing to heterosis here? The first diagram shows the number of primary branches. Again, it seems that there is a small advantage from the F1 as compared to the two parents, but there is no much heterosis in number of primary branches. Whereas, again, length of primary branch a bit up. Yeah, we can say there is heterosis for this one, but more clear one is the secondary branching and as well the tertiary branching. So instead of the primary branch or the length of the primary branch, you may expect that yield advantage may be coming from the number of secondary and tertiary branches. Here you see in the F1, in both cases, you have relatively considerable level of heterosis in both number of secondary and tertiary branches. Whereas in the three genotypes, there was no variation, again, in primary branch, primary length of primary branch, and tertiary and secondary branch as well. So the question is why most of the F1 heterosis was not maintained in F2 population, or it was partially maintained in F2 population. So the first reason maybe is recombination. That means candidate marker was not strongly associated with genes governing the heterosis, or there may be interlocus interaction here. We see that here. We have the candidate locus here on chromosome 4. So this locus may be interacting with another locus here, or maybe it may be interacting with on the same chromosome with another locus. So another suggestion to see is to analyze maybe interaction between locus for this candidate marker. On a separate study, we have identified this the panicle ontogeny of two sorghum inbreds. So around 31 and 38 days after sowing, the two inbred lines were in reproductive phase already. And here you see the branching starting shortly after the transition from vegetative to reproductive phase. Primary branch, secondary branch, and also here initiation of spikelets, as well as initiation of stamen and pistil primordia around 46 and 51st day after sowing in SCB018 and SCB139. So here you see that there is a variation in branching nature, in timing of emerging the different panicle architectures. Let's see also, this is the apex length characteristics of the two founder lines, SCB139 and SCB018. So in both cases, you see a third degree polynomial, initially with a slow growth pattern in both, and eventually followed by a rapid growth in apex length. So this shows the mean apex growth rate on a y-axis and the days in the reproductive phase. Here you see that there is a shift in rank between the two genotypes. So this indicates that in early stage, SCB139 was fast growing as compared to SCB018, but eventually leveled off, and again, SCB018 overtaking the position. So sometimes it may be misleading to judge on the way before the final phenotype is done. So in conclusion, we can say that heterozygosity for the candidate locus had no significant effect, or the effect was very small. So the first suggestion maybe is there may not be close linkage between the target gene and the candidate marker. And whereas there was some tendency of increased panicle weight or dry panicle weight due to heterozygosity at candidate locus. So this may indicate that there is contribution from several genomic regions. So among these, the candidate locus may be contributing for dry panicle weight as well. So F2 populations retained about 25% of the F1 heterosis, whereas the top 50% of F2 and the heterozygotes retained around 60 and 32% of the F1 heterosis, respectively, which indicates that several genes are involved with mediating the heterosis in F1. So probably most important from these findings that is there seems that the heterosis may be due to interaction between locus. That is a kind of epistatic interaction between different locus. This is one of the suggestions. I think we need to analyze further markers, and this, of course, candidate locus may be in interaction with other locus, maybe on a different chromosome or on the same chromosome, but it indicates that there is some kind of interaction between the locus. So we need to analyze further on other markers, like you have seen it, adjacent markers close to the candidate marker for any epistatic interaction with other locus. And also we have also seen from these findings, these studies, that there is variation in timing, nature of panicle elongation and branching, as well as apex growth rate between different founder lines, which suggests that the need for time reference for when phenotyping. Otherwise, it may be sometimes misleading to judge before time. So all of these are some of the references. Thank you very much, and with a special acknowledgement to Imri Ben Israel. He was a PhD student in our lab. Still, he's doing his PhD. Thank you very much.
Thank you, Habte, for giving this nice presentation and completing it just on time. We have some time for questions, please. Yes, Shachal. Let's start up. Microphone? Oh, Moses. Oh, I'm sorry. Yes, please, Moses.
Yeah, Habte. I would like to know the source of your parental lines and then the features of the parental lines that motivated you to select those lines and to study it in this your project. Thank you.
Okay, thank you, Nana. So the source of my parental lines were Israeli core sorghum collection. There were a total of about 200 sorghum collections, which represent all over the world. Then these were also, some of the collections were retrieved from Israeli Gene Bank. So out of these 200 sorghum collections, 20 founder lines were identified based on their heterozygous and based on their diversity and relatedness. After studying for polymorphism using about 51 SSR markers. So these founder lines supposed to be representing most of the sorghum genome. We can say about 200 sorghum collections. So, so this is the source of the parental lines I used in this experiment.
Yes, Shachal, please.
Towards the end of your talk, you presented data on the apex elongation, dynamics of the two parental lines, and you showed that there is a switch in the grading between the two parents. Yes.
Yeah.
Do you know, or is anybody in the laboratory trying to do this kind of analysis also on the F2 population? Maybe this will help to explain part of the yield or reproductive traits, phenotypes that you're observing.
This is not tested in F2 population, but this was basically this experiment was done to compare the inbreed lines or the founder lines against their hybrid. We were including the hybrid as well, but we only presented the the two founder lines, the hybrid is missing because we couldn't verify that it was a true hybrid. It was basically to compare the three genotypes, the two founder lines with the hybrid, and it's not tested in FO2. Most probably we may have similar pattern if we test it in FO2, but
it's possible, but it's a destructive phenotype. You kill the plant. What you do here?
Oh, final phenotype is not. It's not possible. It's destructive type, and we couldn't see the final phenotype, but it's indicative. This is to indicate that early stage measurements sometimes may be misleading, like studying cell division or some activity before, like germination or sealing emergence. This may not be accurate in some case. This is an early phenotype, and that was the idea.
Why is it destructive, Eyal? Can't you do it on one tiller and then, or Habte, why is it destructive? Can't you do it on one tiller and then let some other tillers bear seeds?
That is possible, but it doesn't show the actual phenotype because you have to cut the plant. Once you cut the plant, it may, if you allow the tiller to grow up and take a phenotype from the tiller, tillers also
no, not a phenotype from the tiller. I mean, just to reproduce, for seeds.
We can get the seed, of course, possible. Yeah, possible. That's possible. We can get the seed, but tillers don't represent. Actually, they don't express full of their potential. And
so for doing that, you actually have to destruct several plants at several times and follow up the development.
yes, definitely. Yeah, we have every 4 days we were extracting about 4 plants for every. I mean, it was done for more than about 2 months consecutively. This is a part of that data, and it was a destructive sampling. Yes.
Okay. About the apex growth rate, what accounted for the rapid increase for the 2 lines in this reproductive phase, especially at 16? What accounted for the rapid increase in the blue line?
This is typical of the potential of the variation in the potential of the 2 founder lines, SCB080. So they have a different. I saw some published papers also. If you see the hybrid, if the hybrid was here, you may have seen a clear view of that. So there is variation. If you see the first one, this graph, you have here a slow growth phase initially, whereas usually the hybrids grow very faster in this region, in rapid growing stage. This is from a different study I didn't include in my study. So it is typical of the potential of the founder lines and the nature of growing, accumulating biomass, and that was the reason for the variation between the 2 founder lines.
I have another question, Habte. You were trying to investigate the effect of a specific locus on heterosis. Are you aware of other examples where a single locus have such a profound effect on heterosis to be defined as a major locus of heterotic effect?
Yes, yeah, there have been few examples, like in a study by Krieger et al. Actually, it was from a mutation, but a single gene mutation in a single locus resulted about 60% yield advantage in tomato. This has been published recently in 2010, so it's possible to get a high level of heterosis from a single gene, a single locus. That has been demonstrated, but we can ask how much is the probability from a natural population, how much how can we get that mutation in natural population, or how the approach is a bit different, but it seems improbable to find in other crops also, but it's definitely possible to get a high level of heterosis from a single gene also.
Other questions, Guy? Okay, so let me, please join me. Thank you, Habte, for this